Okay, welcome to this redo of the lecture from Tuesday. As on Tuesday, just want to quickly introduce the team paper topic, which is the molecule known as canstatin. In class, I introduced the concept of a matrikine. What a matrikine is a polypeptide, a peptide or a polypeptide, that is the product of proteolysis of the extracellular matrix. These might be extracellular matrix molecules such as collagen or laminins, fibronectin or fibrin. These are proteins that constitute the proteinaceous fibrillar extracellular matrix. And when they're degraded, what we found is that a number of the degradation products actually go on to live second lives as chemokine-like factors that can stimulate migration, invasion, proliferation, differentiation, and such. Canstatin is one such potential matrikine. So your task is to explore what canstatin is, what the factors are that may lead to its release in a physiological system, and what potential roles it might play in regulating biology. Okay. So what I want to do really quickly is briefly finish up our lecture on the cytoskeleton. Where we left off on Thursday was on this slide discussing cell crawling and the identification of two types of structures, the first known as lamellipodia, which are these sheet-like projections here and here. And pseudopodia, which might be thought of as a structure like this. These are quote-unquote false feet, right, for pseudopodia. Lamellipodia are these lamellar type structures, lamellar shaped feet, quote-unquote feet, that are pushed forward by actin polymerization and other forces, usually during the process of cell invasion. And in this case, these structures are created through actin filament growth, which is being nucleated and occurring at the leading edge of the cell. So we talked about this concept of a Brownian wrench. Okay, so you can see here the formation and the pushing out of this sheet as this cell migrates in this direction. The cell is trying to migrate in this direction. And these are examples of crawling of cancer cells or immune cells. So how does that actually work? It's through a system. One such system is that of ARP23 and WASP. ARP23 facilitates the nucleation of actin growth, and it associates with preformed actin filaments. And so through the actions of ARP23 and WASP, which facilitates the recruitment of profilin actin complexes to a nucleating actin assembly. So not only is it nucleating fibrillar assembly, as you can see here in this example, ARP23 is associating with a preformed actin filament, and as a consequence can nucleate branching points, basically a web-like network structure of actin filaments. And again, this idea of pushing kind of has an asterisk beside it, or should be sort of thought of in quotation marks, because what we believe is really happening is rather than there being a direct force of actin polymerization on this membrane, actin polymerization is occurring on these plus ends, which are abutting up against the plasma membrane. And through Brownian motion, we get fluctuations in the membrane. And these spatial fluctuations due to thermal energy allow for the association, right? The addition of another globular actin on this growing filament. And now, when this membrane is fluctuating, it cannot fluctuate all the way back to this location. It would only be able to fluctuate back to this structure plus another G-actin. So the push is really a prevention of the ability of that membrane to fluctuate back into its original position. So again, think about this idea of pushing at least in response to actin as being in quotation marks. Here's some SEM images trying to essentially pictorially demonstrate the fact that we don't have several pointy filaments that are protruding up against the membrane, but rather this highly complex, web-like, almost mesh-like structure. And again, this is initiated through ARP23 and WASP. Okay, so this gets us all the way back to how listeria gets its actin tail. Listeria expresses a specific protein known as ActA, which is going to effectively hijack the ARP23 WASP system for nucleating actin assembly. ActA has several domains. It has an actin ARP23 interaction site, and it has this proline-rich repeats that associates with a protein called VASP, which is in the WASP family of proteins. VASP, similar to WASP, will recruit globular actin profilin complexes and facilitate nucleation of actin polymerization here off of this ActA protein. The bacterium expresses ActA only on the posterior end, right, the back end of the bacterium. And so you get nucleation of actin filament growth here on the posterior. And through a similar mechanism of this Brownian wrench mechanism, it actually propels, the polymerization of actin propels the bacterium forward. And so again, here is a TEM of the actin tail of a listeria bacterium. So treatments for listeriosis is time for healthy people, folks like you and me. For immunocompromised people, such as pregnant women, elderly, and young children, the appropriate treatment would be antibiotics to kill the bacteria. Prevention is certainly the best way to approach this, and in order to prevent listeria infections, one should avoid soft cheeses made with unpasteurized milk, all the good ones, and avoid refrigerated meat and fish that have not been properly prepared. If you're interested in cytoskeleton and migration, a number of key investigators in cell biology, Jim Casanova, who actually explores ARP23 complex, Doug DeSimone looks at migration during morphogenesis, Dorothy Schaefer in biology explores fundamental mechanisms of cell migration, and myself, among others, in BME. Okay, so with that, we're going to transition back now to molecular motors. Just a few minutes here. Okay. So we're going to couch the lecture on molecular motors in the context of heart failure. Heart failure is broadly defined as when a person's heart cannot pump enough blood to meet the body's need for blood and oxygen, primarily due to decreased cardiac contractility. And this is the point with which molecular motors plays a prominent role. Symptoms of heart failure are dyspnea. We've talked about that before, difficulty in breathing, wheezing, edema, or the pooling of bodily fluids, usually in the extremities. Obviously, if I can't pump blood effectively, then I might get pooling of blood and plasma in certain areas. Fatigue and nausea due to an inability to transport oxygen appropriately, impaired thinking for similar reasons. And as a compensatory mechanism, we'll oftentimes increase our heart rate in order to pump blood through the body. The causes are many. As you can imagine, such a diffuse definition as inability to pump blood might have multiple causes. Certainly coronary artery disease is one. Myocardial infarction, right? So the blockage of blood vessels that leads to a heart attack, death death in regions of the heart. Hypertension, so high blood pressure, which causes an increase in the resistance against our heart. Abnormal heart valves. Hypertrophic cardiomyopathy, so hypertrophic, hypergrowth of the heart. This can be caused by a number of different pathologies, such as even something as simple as the infections around your heart. Congenital heart disease, this means heart diseases that you're born with, such as improper plumbing connections during development, and diabetes. Statistically, about 5 million people in the United States have heart failure per year. About 1 in 9 deaths in 2009, this is about a decade ago, included heart failure as a cause, as a part of the cause. About 50% of people who who develop heart failure will die within 5 years. One of the largest, highest prognostic indicators of heart attack is having a heart attack. So folks that have heart attacks are more likely to have subsequent heart attacks. And the cost to the United States is around $32 billion each year. And I'll point out that this is about the budget of the entire NIH. So we spend more on, or about the same amount, on treating heart failure as we do on all government-based biological, biomedical research. As you can imagine, the prevalence of heart failure tracks with, a lot of times, with demographics and diet. Certain individuals are predisposed to heart failure. Diet plays a significant role, as well as exercise, etc. All right, so let's get into why you need things like cargo transport. So molecular motors, as we highlighted in the pre-lecture, have the sole purpose of generating motion, movement, displacement. Okay, so when we can think about this in two contexts. One is motion of bodies, right, of a human walking from one place to another. The other is motion of intracellular compartments, which is really where motor proteins work. So if we didn't have cargo transport, or what we would call active transport, then we'd rely completely on things like diffusion in order to get two molecular species together. And so we've talked a lot about degrees of freedom and bringing proteins, restricting proteins, and the distances that they have to diffuse by tethering them to membranes, right, to regulate, to force them into 2D diffusion, to the creation of things like lipid rafts, right, that further restrict the distances with which proteins are within proximity of their partners that are required for signal transduction. Transport, or active transport, is another way that we can reduce the time with which, or the rate with which, two proteins, two molecular species might interact with one another to catalyze a type of reaction. Right, so below is a simulation of a random walk of a diffusing particle over time. And you can see this tortuous path that this particle is taking with respect to time. So if the goal is to get it from point A, here at the bottom, to point B, here at the top, the path that it's taken is not the most efficient path, right? And so this is what diffusion will give us. So let's do a really quick calculation and talk about what are the equations used to calculate how long it takes to diffuse. And for spheres, we use what's known as the Stokes-Einstein equation. This is Navier-Stokes and Albert Einstein. This is work that they had performed during their graduate studies. And what this equation here on the right basically says is that the mean square displacement, so if we don't consider the fact that a particle might move, let's say, one step to the right, let's say that's positive direction, and then immediately one step to the left, let's say that's a negative direction, if I add those up, not as an absolute value, right, but as discrete steps, and I would say +1 - 1 equals 0. So it's diffusion, or it's the distance traveled, the displacement is 0. But in fact, that particle has moved 2 spaces, right? One to the right and one to the left. So that's what this term is meant to do, is to add up all those different displacements. And this is proportional to, this equation is definitely something you want to know, right? It is in blue. More importantly is understanding the relationship between the displacement of a particle and these various factors. So let's look at N. N is a value of dimensionality. So this is a value that's placed in the numerator of that equation based on whether or not the particle is diffusing in one dimension, meaning along a line, within a plane, or in three-dimensional space. KBT is the Boltzmann's constant, right? Which is a relationship between the amount of energy per unit temperature, right? And then times the temperature, right, in Kelvin. Okay, this is the absolute temperature in Kelvin. Okay, in the denominator, we have 3 pi, and we have 2 other variables. One is the viscosity of the medium. Obviously, a particle diffusing in water will diffuse much faster than in a more viscous material like, say, molasses or honey. R then is also the radius of the sphere. So the larger the sphere, the smaller the displacement is given all these other variables, right? So it's proportional to the dimensionality, how many degrees of freedom that particle has to move, and the energy in the system. KBT is the energy in the system, right? It is inversely proportional to how viscous the media is and how big that particle is. So, just for your information, the bulk viscosity of the cytoplasm is about 1,000-fold more than that of water. So if we do a calculation of the diffusion, right, and the mean displacement of a particle along the length of what is a fairly average axon, about 1 meter, right? That's going from your brain stem, let's say, to a muscle that it wants to stimulate. If we're trying to transport a ribosome, right, from that, from the perinuclear, right, the region right around the nucleus to the end of that axon, it would take on the order of 700,000 years in order for that ribosome to diffuse down the length. Okay, so the take-home message here is that diffusion is not sufficient for biology, certainly as we know it. Okay, so in comes molecular motors, a method to input more energy, chemical energy, and convert that into actual work and displacement. Okay, so molecular motors are enzymes whose primary function is to convert chemical energy, chemical energy into mechanical work. Work expressed in the units of joules is the amount of force generated times the displacement. Work equals force times displacement. The chemical energy that we're going to use in biology comes from 3 different molecular species. The first is adenosine triphosphate. We've talked a bit about that. Guanine triphosphate, so ATP and GTP. And remember, the hydrolysis of one of those inorganic phosphate groups from ATP to ADP plus inorganic phosphate, or GTP to GDP plus inorganic phosphate, the breaking, the hydrolysis of that bond relieves, right, energy. Another source of chemical energy can come from electrochemical gradients. We won't talk a lot about that. Again, motion is always the end goal, either in the motion of actin filaments across one another, or myosin filaments and actin filaments in the context of cell contraction, motility of a cell, or in the transport of cargo. All right. So motors are matched to their tracks, right? And so we have certain motor proteins, the kinesins and dyneins, that will only associate with microtubule motors. Kinesins move in the positive direction, and dyneins move in the negative direction. Excuse me. Oop. My mouse is getting a little touchy today. Actin motors have myosins, and in the pre-lecture we described the heterogeneity within the myosin family. Lots of different proteins, structurally all a bit different, and likely have the structure leads likely to the diverse functions that myosins play. There are no motors that run on intermediate filaments. Why not? The reason is that there's no polarity, right? There's no directionality to intermediate filaments. And so in the case of microtubules and actin motors, right, there's always a plus end and a minus end, right? There's some directionality so that these motor proteins like kinesins can operate, move in one direction or another. On intermediate filaments, because of the randomness with which they form, there's no polarity, thus no molecular motors. Microtubules and actin filaments themselves are, again, sometimes considered motors because they can, quote-unquote, push on things as they polymerize. And we saw one demonstration of that when we talked about membrane protrusion, such as lamellipodia, as well as the listeria bacterium. Okay, so let's talk a little bit now about regulation. How are these things regulated? And their basically regulation is associated with keeping the motor protein, right, the molecular motor, in an off position. Okay, and there are two different approaches. The first is that the motor itself might be on, but we can actually block the ability of the motor to bind sites on actin or on microtubules. This is going to be the case in that of striated muscle myosin. So that would be your skeletal muscle and your cardiac muscle. And we'll go into that regulatory mechanism here in a minute. The other is to actually modulate the activity of the motor heads themselves, right? So we can make the motor heads then unavailable for binding by regulating their conformation. And this is pretty much everything else. Okay, so let's take that first one about blocking motor binding sites on actin. And I mentioned that it's associated with striated muscle. And striated muscle is that of your skeletal muscles, right? The ones that help you grossly move your arms and legs, and cardiac tissue. Striated muscle gets its name from the different striations, both here you can see in the vertical axis as well as in the horizontal axis. There are striations that can be observed through light microscopy. And regulation of contraction of striated muscle is accomplished again by inhibiting or allowing the binding of myosin to actin. In this particular case, there's a broad inhibitory activity associated with a protein complex, and this is relieved by things like calcium. This is a concept we call disinhibition, not activation, right? So we're not physically activating actin, we're not physically activating myosin. What we're doing is we're inhibiting an inhibitor. And so you'll oftentimes see this particular language of a disinhibition, right? It's a double negative almost, that leads to a positive event. This is based predominantly, this mechanism is based predominantly on the thin, what's called the thin filament. So let's look really quickly at some of the anatomy of a striated muscle. The primary force-generating unit is known as the sarcomere, and it's comprised of a Z-band, here described on the two ends here. That would be here, these dark regions, right, here, here, and here, okay. An M filament, an M line, right, which is this central line. And then we have what's known as thin filaments, which are actually actin. So these are actin, actin filaments. They're always oriented with the plus end towards the Z-band. What this means is that this actin is fairly stable, right, because the polymerization of actin occurs predominantly on the positive end. Again, remember, due to the high critical concentration at the minus end of the actin filament. Okay, so many, many, many actin filaments are going to be connected through this thing called the Z-band. Myosin 2 is also going to form a filament. This filament we call the thick filament. And again, it gets this name, the thick filament, because it is the thicker of the two filaments when you zoom in to this structure. Remember, in the case of myosin 2, it has a head domain and a very long tail domain. And the tail domains self-assemble into this polymer structure. And they're connected again through this M line. Right? Just as an anecdote, a little bit of trivia for you. This protein here that we won't talk about is a protein called titin. It's a very interesting protein that's comprised of what's known as fibronectin type 3 repeats. We'll talk a little bit about that in extracellular matrix. These are really interesting elastic springs, elastic spring domains that unfold and refold with exceedingly small amounts of energy loss. So they are essentially nature's perfect spring. And that sort of connects this whole apparatus together. The final piece of vocabulary that I want to focus on is this thing called a crossbridge. And the crossbridge is just as it sounds. It's the region with which the thick filaments and thin filaments cross, right? So it's this region here. Okay, great. All right, so let's talk a little bit about regulation. Skeleton muscle filaments, we talked about them here. They're sort of very basically schematically represented as an actin filament. But as you'll find as you dig more and more into biology, there are additional levels of complications and complexity in the systems. And the thin filaments are no different. They're associated with a protein known as tropomyosin. It's this pink protein here that wraps all the way around. And a complex known as the troponin complex. It's comprised of 3 proteins known as troponin C, troponin I, and troponin T. These are the regulatory element that modulates whether or not these myosin head domains can interact with actin filaments or not. Okay. Now, in the case of skeletal muscle, there's essentially no real regulation of the myosin head domains themselves. But I'll go ahead and introduce the additional light chains, so-called the essential light chain and the regulatory light chain of myosin in this context, because they'll come up when we talk about all other forms of regulation. The essential light chain, the ELC, is essentially a structural protein that positions the head domains in a certain configuration. And the regulatory light chain is just what it sounds like. It's the region of it's the subunit or the chain of myosin that will facilitate the activation of myosin. So let's dig into the tropomyosin complex. Tropomyosin, in and of itself, does not inhibit contraction. What it's doing is it's inhibiting the ability of myosin to bind to actin. Okay? All right. So the troponin complex, again, is those 3 proteins, TNT, TNI, and TNC, or troponin T, troponin I, and troponin C. Troponin T is the tropomyosin binding unit. Okay, so this is going to bind to tropomyosin. Troponin I is called the inhibitory subunit. So this is what's believed to do most of the inhibition, or at least positioning, and we'll find out here in a minute, positioning tropomyosin and TNT to inhibit the interaction. And TNC is a calcium binding unit. Okay, so the C comes from calcium binding, the I comes from inhibitory, and the T here comes from tropomyosin binding. So TNT and TNI together with tropomyosin inhibit the myosin binding to actin. If we look at a crystal structure, in this case, we're looking at a surface map with an interposed ribbon structure here. You can see where the calcium is going to bind, and you see this basic complex. TNC, here's the troponin, TNT and troponin. Here's the inhibitory complex, and TNC. All right. So let's say you have nervous stimulation because you want to contract a muscle. Nerves stimulate. They stimulate through the release of neurotransmitter at the neuromuscular junction. We're not going to talk about that in this class, but they'll induce a large efflux of intracellular calcium stores, normally in the endoplasmic reticulum. is come out of the ER very, very quickly into the cytoplasm, raise the calcium concentration inside the cytoplasm, and upon that calcium influx, TNC is going to bind calcium. When it binds calcium, it's going to undergo a significant conformational change. And there are two theories about how this is transmitted into a disinhibition impact. The classic theory is that this conformational change in TNC is quote-unquote transmitted into TNT and TNI to actively move. So that would be the key here, is it's actively moving tropomyosin off the myosin binding site. Okay, so let's pretend that these little blue dots are where myosin will bind to actin. The tropomyosin and troponin complex here are blocking that site. You get calcium, and calcium causes a conformational change that actively pushes that structure off the sites, and now myosin can bind. There's a newer theory, which is in all likelihood more correct, which is that TNI locks the tropomyosin to actin, and when calcium is present, conformational change in TNC allows TNI to unlock tropomyosin, and now tropomyosin is free to diffuse. Now, it's not going to diffuse fully away from the filament. It's going to undergo these sort of Brownian fluctuations, right? So it's going to wobble and wiggle about. And in doing so, it avails the myosin binding domain, right? So it provides an enhancement in the capacity or ability for myosin to recognize and to see that site. And then cooperative binding by myosin then completes the process by stabilizing the dissociation of tropomyosin away from the myosin binding site. So you can think about it like this. It's wobbling around, myosin binds, and then it stabilizes that displacement. So we can summarize that entire pathway in 4 bullet points. So calcium is released from intracellular stores, usually due to neural stimulation. Calcium then binds troponin C, causes a conformational change, and then tropomyosin is unlocked from the myosin binding sites on actin. The mechanism is unknown. There are theories, as I've just described, but the mechanism is actually, as of such, undefined. Myosin is now free to bind. Okay, so we've just talked really quickly about the regulation of how myosin would bind thin filaments or actin filaments in striated muscle. Let's talk now about the actual force generating and displacement generating component of these structures. And that comes through the actin-myosin crossbridge cycle, right? So remember that crossbridge is that region with which myosin is capable of interacting with actin. This cycle refers to the conformational changes that myosin head domain undergoes in order to generate steps or walking or force. So we're going to start here in the bottom right-hand corner, and this is really what we're going to call step 1. Okay, you could pick any step. We're going to call this one step 1, and there are some reasons. This state right here, where myosin is in this bent this bent conformation, myosin is bound to ADP, adenosine diphosphate. The inorganic phosphate is long gone, and we call this the rigor state. The reason we call this the rigor state, rigor in Latin stands for stiff or stiffness, right? And this comes from the term rigor mortis, so stiff after death. And it gets its name because when you analyze muscles via autopsy, right, following death, the myosin is in this particular configuration, and so the muscles in this state, when they're all in this state, are stiff and rigorous, right? Okay, so that's the rigor state. Myosin bound to ADP, and it's strongly bound to actin. Okay, this particular conformation of myosin is has very strong affinity for actin and will not let go, or does not let go easily. Just to remind you and orient yourself, in this particular actin filament, this is the plus side, this is the minus end, and so the myosin is moving in this direction, right, in the plus direction. All right, so step 2, ADP is released, okay, and notice the curves, right, or excuse me, the arrows. Strong arrow going in this direction, weaker arrow going in this direction. It shows the reversibility of this, right? So ADP is associating and dissociating, but there's a much stronger force, right? Much stronger constant, chemical constant, or rate constant, right, for ADP to be released than for it to be re-added to this species here. So step 2, ADP is released. Step 3 and 4 are in this sort of weak zone, right? So the first, step 3, ATP is going to bind to myosin, and myosin's going to release. Okay, that's step 3. It's binding and releasing. Okay, so ATP is a weak binder for filamentous actin, right? So for F-actin. So the loading of ATP weakens that bond and it'll let go. Okay, step 4 now is this process by with which the ATPase activity of the myosin head domain, or of the myosin, excuse me, is in the process of relieving, right, of hydrolyzing that inorganic phosphate to form ADP, from ATP to ADP plus inorganic phosphate. Okay, so ATP hydrolysis cocks the motor domain, right? So it goes from this conformation here into this more linear or straight conformation. This is called cocking the myosin head domain. So the motor domain, and now it's going to rebind. But again, this species here is very similar, right? It's in this state of flux. It's in this intermediate state where it has ADP, but it still has that inorganic phosphate sort of associated. So it's a weak binder, but it changes the binding affinity, right? The release of this, or the hydrolysis of this inorganic phosphate increases the binding affinity for the species. It's now going to bind. And that leads to step number 5, what we call the power stroke. So ADP plus inorganic phosphate is now rebound to the actin. And now when it ejects that phosphate group, the energy from ejecting that phosphate group goes into a conformational change where the head domain goes from looking like this to looking like this. And that's where the step occurs. This is called the power stroke. So the motor domain takes a step, and inorganic phosphate is released. And we're back to this rigorous state. Okay. The net result of all of these individual interactions, right? We're talking about this interaction right here when we describe this cycle. That's occurring here, 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 all along this structure. And remember, muscle is made up of many, many, many sarcomeres. So the net result of this, because our positive ends, right, are here and here, these guys are walking in this direction, these guys are walking in this direction. As a consequence, the entire sarcomere shortens, right? And this is muscle contraction. This is done primarily through this process known as fiber sliding. In this case, actin fibers are quote-unquote sliding against myosin fibers. There is one other example that I want to highlight on sliding filaments generating motion, and that's in the context of microtubules in flagella. Flagella and cilia are moved by these things known as actin-myosin, which form similar types of structures with microtubules that we've just seen with myosin and actin. Oh, excuse me. And as a consequence of those power strokes, right here, coordinated power stroking of those complexes leads to this sort of gross motion, okay? And then there's a recovery stroke. That's observed sort of mechanistic, a little bit more mechanistically here, where you get these sort of bending moments around these fibers that are sliding against one another because they're anchored in one direction and moving in the other. So this is sort of like almost like a thermostat, right, where you have two metals against one another, and due to their various expansion coefficients, right, you get bending. That's what's happening here. Okay, so let's now talk about regulation of contraction in smooth muscle cells. And this type of regulation is going to also occur in non-professional contractile cells, such as fibroblasts, myofibroblasts, these types of things. This is more representative of how most of the myosin types are actually regulated. And it's based predominantly on myosin, not in the thin filament. Okay, so we're going to regulate the availability of those myosin head domains in order to bind actin, as opposed to inhibiting actin, the myosin binding sites on actin. It's accomplished in smooth muscle cells by phosphorylation of what's known as the regulatory light chain of myosin. We've talked about phosphorylation before. This is a kinase, is an enzyme that will phosphorylate. And in the case of that regulatory light chain of myosin, which I highlighted a few slides ago, the kinase is called myosin light chain kinase. That's the name of that kinase. Okay, so in smooth muscle cells, we also have these things known as thick filaments and thin filaments. They're just less robustly structured than they are in skeletal muscle, where we have bundles of myosin head domains, right? Bundles of myosin that are going to contract or walk, right, in these various directions, causing sliding of these thin filaments and ultimately contraction. Phosphorylation is induced indirectly by calcium, right? So, and I highlighted for those that were in lecture, remember that I went off on a little bit of a soapbox tirade that there are also kinases that are calcium independent. So yes, phosphorylation can be induced indirectly by the activation of kinases through calcium, changes in calcium. But there are also kinases such as integrin-linked kinase, which directly phosphorylate the regulatory light chain of myosin that are completely independent, do not require calcium in order to operate. Okay, so the regulatory domain of myosin, let's highlight that a little bit. Here's the cartoon, right, the schematic that was shown in the pre-lecture. Smooth muscle myosin is activated by phosphorylation of the regulatory light chain. So if I take this structure and I'm going to zoom into this region right up here, right? So the motor domain in this picture is way down here. Here's the regulatory light chain. Here's the essential light chain. If you remember about the essential light chain, this is a structural protein that helps position things in the right orientation. And now we're going to get a phosphorylation event. There's the essential light chain, regulatory light chain. We're going to get phosphorylation events on threonine 18, so that's the 18th amino acid in RLC, and the serine, which is the 19th position. Right, so threonine 18, serine 19 will be phosphorylated by those myosin light chain kinases. These phosphate groups can be removed by phosphatases known as myosin light chain phosphatase. And it's the presence or absence of these phosphate groups on these two structures which position the head domain such that it can bind actin. So the regulation of phosphorylation requires both the kinase to add those phosphate groups and a phosphatase, which is going to remove those groups. And the balance between the relative active kinases and active phosphatases that are manipulating the site will yield the overall activity of myosin in that cell. So how this works, don't take this cartoon too, I'll say too specifically. But what's going to happen is that in the inactive state, the head domains are positioned in an orientation that does not facilitate their binding to actin. We get phosphorylation events, and those cause conformational changes to the overall structure, which then reposition the myosin head domains in an orientation or configuration that allows them to bind actin. Again, phosphorylation events cause conformational change, something we've heard a lot about already, leads to activation of myosin. There are other examples, in this case of kinesin, where cargo rather than phosphorylation can cause these events. So in this case, you have a vesicle. The vesicle binds the cargo binding domain, and that binding of the cargo domain to its cargo yields a massive conformational change which positions the motor domains, right, the head domains of the motor protein in a configuration to bind, in this case, microtubules. We discussed this in class, but just as a reminder, this is allosteric regulation, right? The cargo is binding a site that's distant from the activation, right, the enzymatic domains, causing an activation state. Allosteric, action from a distance. Okay, so what happens in heart failure? The reduced myocardial contractility is because one or more of the following. A, we can lose cardiac myocytes, right? So if we have less contractile cells, we get less contraction. We can change tissue integrity. So let's say we through unknown mechanisms, wind up undergoing fibrotic remodeling. So we get scar formation in the heart. This causes an increase in the resistance against those muscle cells that are trying to contract. We can have reduced availability of ATP, right, the chemical energy source, in order to generate the work. We can have impaired calcium recycling. Right? Again, it's this calcium efflux that causes this coordinated contraction amongst cardiac myocytes. Or we can have sarcomeric abnormalities. Regardless of the cause, we get a decrease in the contraction of the heart. So along comes some pretty clever scientists that discovered this drug here. I'd love to say I know how to say it, omecamtiv mecarbil. I think that's right. And what they decided was, instead of trying to regenerate cardiac myocytes, which everyone's trying to do, and instead of trying to eliminate the fibrosis, which we still don't really know how to do, what happens if we can actually treat what's remaining, right? So if you take the remaining cardiomyocytes that are functioning, what if we can turn them into super cardiomyocytes? One way we could do that is to increase the rate of force generation by altering myosin-actin binding kinetics, okay? So we're not going to change, in this particular case, I want to remind you that what this drug is going to do is increase the rate of the actin-myosin crossbridge cycle, all right? Those are occurring at the molecular level, not at the cellular level. So the theory behind this is that by increasing that on rate, right, that cycling rate, that we can get more the similar types of force generation, or excuse me, I should say this differently, more force generation per cell, okay? So what omecamtiv mecarbil, or OM, does is it binds cardiac myosin, and allosterically, talked about that word just last slide, increases the transition rate of myosin to its force generating state. Hopefully you remember what that force generating state is. That is the power stroke. So what it's going to do is it's going to work by decreasing the energy barrier between this weakly bound state and the strongly bound state, right? So we're going to increase the rate that we move through steps 3, 4, and to 5, okay? So by increasing that rate, we increase force per cell. What the result is an increase in overall contractility while keeping the pacing of heart, or the heart rate, the same. What this does is that it increases cardiac output, right? More force per muscle. Now, in this particular case, it's hard to tell. It does look like this guy is beating faster, but statistically, he is not beating any faster than this guy. But what you can appreciate is the amount, if this is the lumen, one of the lumens of the heart, one of the heart chambers. What you can definitely observe is that this guy is pumping, right, is squeezing out more of that blood that's filling this cavity than this, right? So that equates to more blood being moved per pump, right, per contraction of the heart. If you think molecular motors and heart failure are cool, then I would strongly suggest you go and talk to Jeff Saucerman in our department, also Will Guilford here in biomedical engineering. And with that, I thank you for your patience while I have re-recorded this lecture due to technical difficulties. I'll be posting this soon, and I look forward to seeing you guys on Thursday. And everyone be ready for the quiz. Thank you.